Greetings and welcome. So today we're going to be talking about sex-linked traits and reviewing the pedigrees that you worked on yesterday, and then we're going to be moving on to dihybrid crosses. So sex-linked traits. Here's basically the answer key for what you did yesterday. Kind of looking at this, how do we know that this one woman right here, this is Queen Victoria, how do we know that she was a carrier? Well, there's no father listed, but it shows her son has it, and so therefore we know that she was a carrier. So we look across here, and this female right here, we don't know. Could she be a carrier? Well, she got a normal X from her dad. She could have got either of them from her mother, so we don't know for certain. Sorry that the H's are not subscripts. They should be, but the program I was using to edit this just wouldn't do a subscript. So anyways, I would fill in all the males that are blank as XY and all the males that are filled in as XHY. All right, so you should have that done. And then the females go back and fill in and then just look. All right, so for this one, does she have a father that has it? Well, not. You don't know. Does she have a son? Yes. Moving on. Next one. Does she have a father that has it? No. Does she have a son that has it? Yes. She has to be a carrier. No children? Possibly, because mom was a carrier. All right. This female. Now, she married in, so I'm just going to go ahead and assume that she's normal and not a carrier. That's usually a pretty safe assumption. Technically, it's probably XX question mark. You don't know for sure if she's a carrier or not because you don't know anything about her family history. But, again, marrying into the family, you can safely assume that. All right, here we have XXH. Why do we know that she's a carrier? Well, she does not have a father that has it, but she does have two sons that has it, so she has to be a carrier. This one right here. Her mom was a carrier. That doesn't help. Father didn't have it. All right, that doesn't help. But she has two sons that has it, so we know she's a carrier. Could these two both be carriers? Well, they could be because mom, again, was a carrier. So these guys could be carriers. We're not certain. No children were produced. Can't tell. Going over this female, same story as the previous ones. This female, father doesn't have it, but the children do. One of the children does, the son. So therefore, we know that she has to be a carrier. All right, moving on over to this female. She has a son that has it, and she has a father that has it. So I tell you to look at the father first because that's easier. Well, the father has it, so she has to be a carrier. But son has it too. So moving along, next female's right here. Father doesn't have it, but sons do. She has to be a carrier. Going along this row. While mom was a carrier, so she could be a carrier, and same story with all these. No children were produced. The Bolshevik Revolution ended all of their lives. So if you kind of look into the Russian royal history and their families, yeah. Anyway, that's why there was no children produced. Same thing here. Again, mother was a carrier. She could have it. She could be a carrier. We don't know for certain. And then same thing with these two right here. Mother was a carrier, could have it. Matter of fact, had brothers that had it. Had a lot of brothers that had it in this particular line. From whom did Leopold, that's number 3, inherit his hemophilia? Did he inherit it from his father? No, because he got his Y chromosome from his father. So he inherited it from his mother, so from his mom, or Queen Victoria. What is extremely unusual about Leopold III? Or Leopold, number 3. Well, he had hemophilia. He didn't have normal blood clotting. So what are the chances that Alice of Hesse, number 2, so that's right here, or Beatrice, number 4, being carriers? They have to be carriers. We know that they're carriers. We figured that out from just filling the whole thing out. What were Leopold's chances of passing hemophilia on to his son? 0%. Leopold cannot pass it on to his son. It's impossible because he has to pass the Y chromosome on to his son. If he could pass it on to his son, he'd pass on the X chromosome, and his son wouldn't be his son would be his daughter, if that makes sense. So, yeah, it's his son, had to get a Y chromosome, not possible. Under what circumstances would it be possible for a father and a son to be hemophiliacs? Basically, mom has to be carrier, and it has nothing to do with the father having it and the son having it. Looking at what was turned into me for homework, a lot of you guys put H's here. No, these are sex-linked traits, so you should have XXH and XY. And here you go. This is the way to put it. Square should be filled out. The possible genotypes are these four here. 50% of females are carriers, but 100% of the females are normal. 0% are hemophilia. 50% of the females are, 50% of the males are normal. 50% have hemophilia. We look down at this bottom Punnett square and we switch over to C's because we are now looking at color blindness. Again, fill in all the males. If the male's shaded in, he's XCY. If he's not filled in, he's XY. Then start going through the females. All right, does she have a father that has it? Don't know. Does she have a son that has it? Yes, has to be a carrier. She married into the family, so I'm going to assume completely normal. Does she have a father that has it? No. Does she have a son that has it? Yes, she has to be a carrier. Does she have a father that has it? No. Does she have sons that have it? No. Probably normal, but could she be a carrier? She could be. So so this one definitely has to be XX question mark because mom was a carrier, so she definitely could be a carrier. Not likely. I mean, you got a decent sample size here, but again, not big enough. I mean, really, you'd have to have a son that would have it to prove it. And she's only got 2 kids, and that's kind of like analogous to flipping a coin heads up twice in a row. Is that probable and possible? Absolutely. Like, that's not a far stretch. So, you know, unless you saw some children down here somewhere that had it, then you would not be able to know that she's a carrier. All right. And sometimes you can get a situation like that. Like if this daughter had a son that had it, then you would say, all right, I know she's a carrier. And if she's a carrier, that means she got it from her mom. So then we would now know mom's a carrier. It's a little higher level and a little bit more than what they usually typically would ask you on the end of course exam. But you do have to actually be able to look at that. I just tell you, look at the mom and for the woman, look at the father and look at the sons. But sometimes you do have to look down a couple of generations and think about it as you fill it out. Like when you sometimes it's kind of beneficial to kind of work your way back up on these. I know I start from top to bottom, but if you do start at the bottom, sometimes that does help you because you can work your way back up. Like, for example, we know that this guy has it, so this woman has to be a carrier, and because she's a carrier, well, her father has it. So but if these were swapped, then we would know that mom was the carrier there, okay? All right, so the rest of the females here. All right, so we know she's a carrier. How do we know she's a carrier? Because she has a daughter that has it, not a son. Well, then how does that? Well, because the only way a woman can have it is if dad has it. Well, dad does, and mom's a carrier. So that's the only way this individual 3-13 could have it. So that's another way of proving that she's a carrier. So a lot of this is just thinking it through. Okay. This last part trips up a lot of students. So take a look at this and think about it closely. These are really good examples of questions you'll see on either the end of course exam, ACT, SAT, things like that. All right, so what's the odds that number one being a hemophiliac if mother was? So if we think about this and we draw a line up here and we have a mom, all right, and mom is a hemophiliac, then she has to, where did he get his Y chromosome from? He has Y chromosome from dad, so where did he get his X chromosome from? From mom. Well, if mom is XHXH, the only option he has is to get an XH from her, and since the Y doesn't really mean anything other than you're a male, then we know 100% for certain that this person is a hemophiliac. Like if mom was a hemophiliac, he has to be a hemophiliac. Of number 2 being a carrier for hemophilia if father had it. So we're going to assume that anyone else that's not mentioned is going to be normal, homozygous normal. So what's the odds that she's a carrier? Well, if dad had it, he was XHY. She got her X chromosome. She got one from mom, but she also got one from dad because she has 2 X chromosomes. If she had a Y chromosome, she'd be a boy. So it's a female, it's a circle. So therefore, since it's a circle, we know that she is a carrier, 100% for certain. Number one being a hemophiliac if his mother's father had it. So this is 3 generations that you need to think about it. So his mom had a father that had it. So what does that make mom? So if his grandfather had it, he was XHY. So that made mom a carrier. So if mom's a carrier, what's the odds of him having it? Well, so mom's XXH. So he could either get a normal X or an XH. And that means that he has a 50% chance of having it. So for number 10, 50%. Number 11, number 1 getting hemophilia from his father. Remember, where do sons get their hemophilia? They get it from their mother, not from the father. They get the Y chromosome from their father. So anytime you see anything about a son getting it from a father, it's impossible. It's always 0%. Number 2 being a hemophiliac if her father was one. Well, if her father was a hemophiliac, that doesn't make her a hemophiliac. Yeah, she got one bad trait from dad, but we're going to assume that mom was XX, just normal. That makes her a carrier, not a hemophiliac. Number 13, number 1 being colorblind if his father was. Again, fathers and sons are always 0%. Number 2, giving hemophilia to her sons if she is a carrier. All right, so if she's a carrier, so she's XXH, right? So she's going to pass on, she's either going to pass on a normal X or the XH. That means it's a 50-50 shot of a son having her bad X chromosome or her good chromosome. All right, I'll go ahead and post this key. If you have any questions, feel free to ask. All right, so moving on to today. Today we're going to talk about dihybrid crosses, and we're going to start to get a little bit more into how genetics actually works, a little bit more. If you remember, I've said on multiple occasions that if you ask me about eye color, I'm basically going to lie to you and not tell you exactly how it works, because honestly, it's really, really complex. It's not just one gene. A lot of times we look at one gene when we talk about eye color and we say, all right, we model it with complete dominance, that brown eyes are dominant over blue eyes, which is basically more or less true. And then we look at incomplete and codominance, and that gives us 3 eye colors. And again, that's kind of true, but really it has to do with multiple genes on multiple chromosomes. And so it's not just one. All right, so here are the learning objectives for this lesson. And the big thing here is polygenic traits. So a lot of our traits are determined by different genes on different chromosomes, and they kind of work together to produce what our final phenotype is. Describe the effects of pleiotropy and epistasis. We're going to get more into epistasis at a later time. For today, it's going to be more of a focus on the polygenic traits. All right, so pleiotropy. Pleiotropy is a single gene having multiple phenotypic effects. So if you guys are familiar with Siamese cats, I have a Siamese cat, notice that the color points at the ends of the ears, the face, basically where its body's the coolest. And so what happens is this gene produces this protein, and it denatures or breaks down when it's warmer. As a matter of fact, I've had Siamese cats before, and the one would sit on the heating register, and he'd have almost like a little square patch on his belly in the winter from the warm air blowing on his gut and making it whiter, because it actually breaks down the protein as it's warmer. So its body temperature is basically warm enough that it denatures this dark pigment. Now, what is this with pleiotropy? A single gene can have multiple phenotypic effects. In other words, yeah, it produces this pigment that makes the points the darker color and not its body color. However, what also happens is, notice this cat kind of has these crossed eyes. And that's because the same protein is used in the manufacture of these tendons that are in the eyes that basically make the muscles a little weaker. And so a lot of Siamese cats have these crossed eyes. And it turns out that it's the same protein, just used in a different structure in a different body part. So we see this actually quite a bit. So that's one pleiotropic trait. They're both caused by that same gene, and it causes what seems to be two different traits to actually be together. They seem to be unrelated, but they actually are caused by the same gene. Or, well, same allele of that gene. So here we have albinism in rats, and all right, so they're white, they have these red eyes, but it also makes them very, very sensitive, and actually their eyes, a lot of times they are blind. A lot of white rats are blind because the sunlight actually damages their eyes, and you might think that, you know, blindness and having white fur are unrelated, but it's not. Another example of this is white cats are frequently deaf. Again, same protein that makes their fur color white does something with their inner ears and makes it so that they can't hear. An example in humans is Marfan syndrome. A lot of NBA players are extremely tall, and one of the reasons that some of them are tall is because they do have what's known as Marfan syndrome. And what it does is it makes it so that your connective tissue is a little looser, and it makes them have these really long limbs, which makes them a lot of times really tall, which is great for being an NBA player. But also notice that a lot of NBA players, one, because of the height and just like the angles and pressure and such, they suffer a lot of times from like a lot of foot and ankle problems. Again, part of that's being athletic, but also part of it's also just being tall and putting more pressure on her. But some of them have really, really weak ligaments because of this protein that's not formed properly and makes their ligaments extra stretchy. And so a lot of times they have problems with those joints not holding together as strongly. Well, because they don't hold as strongly, a lot of times they have larger hearts, which, I mean, a large heart, you might think, well, that means it's going to beat harder and therefore, you know, make you a better athlete. But what happens is a lot of times you will have these blood vessels in the aorta where it's got this connective tissue, and it actually is weakened and can bulge, and if this bulges, it can explode. So there, again, this is only a select few in the NBA, but it's disproportionate in comparison to the rest of society, really, because again, their disorder has made them longer arms, longer limbed, and therefore taller, but also gives them this propensity to have these aneurysms, or basically their aorta more or less can explode. And if that happens while they're running around, which again, NBA players are going to run around, that's life-threatening. I mean, they would pass away before they hit the floor, really. So there are some NBA players who are suspected of having Marfan's or do have Marfan's, and they go in for frequent heart exams because heart problems are, again, disproportionate compared to the rest of the population with NBA players. So again, that's a pleiotropic effect. Again, tall and thin, long extremities, but again, problems with their eyes, skeletal system, and then enlarged heart. So when we look at dihybrids, we're going to look at two traits at the same time. And so we're going to look at, notice that the B's go together and then the C's go together. And so basically we're looking at two genes at a time. So the probability of receiving two recessive alleles from a heterozygous parent is 1 in 4. All right, so how do you figure out probability? So just think of it this way. What are my odds of flipping a coin and getting heads? Well, it's a 1 in 2 chance, right? So if I flip the coin, it's either going to be heads or tails. That's my two options. The one I'm looking for, the desired outcome, is 1. 1 out of those two are heads. Well, what about getting heads twice in a row? Well, if you think about it, there are 4 options if you flip a coin. 2 coins, sorry. If you flip 2 coins, there are 4 options. I flip them both, and they're both heads. That's one option. I flip them both, they're both tails. That's another option. Now, let's assume that one coin is shiny and one's a little bit more dull. And so we flip these coins, and that's going to be a difference because the shiny one's head and the dull one's tails, or it's the other way around, where the shiny one's tails and the dull one's heads. See how that's 4 different options? And so you could actually have 4 different possibilities. And if we want them to both be heads, that's 1 out of 4. So how do you get that? Well, you multiply it. 1/2 times 1/2. When you are taking 2 probabilities and you multiply them together, that gives you the probability of having both of them together. So here's another example. Let's just say simple complete dominance. Let's say blue eyes is recessive, right? So in my family, both me and my wife are carriers for blue eyes. We both had a father that had blue eyes. Neither of us have blue eyes. What was the probability of having two children with blue eyes? All right, so probability of the first child having blue eyes is 1 in 4, right? Because if you do the Punnett square for that, you would have, we know that my wife and I are both heterozygous for the trait. So capital B, lowercase b, capital B, lowercase b, and we're going to run this cross. All right, so this is not blue eyes. This is, let's say, brown eyes. Brown eyes. Brown eyes. And here we have the blue eyes. Now, here's the major misconception that a lot of students have, that this one right here represents one child. And my first child did, sure enough, have blue eyes. Okay. So I said, what's the probability of both of them having blue eyes? Well, some people assume that this represents a child. No, this represents a possible outcome of the genetic combinations of the parents. So it's a 1 in 4 chance of getting this, but it's a 1 in 4 chance every single time you have a child. So the next child doesn't have a higher percent chance of getting these other traits. Again, it's kind of like saying, all right, I won the lottery ticket today. I had a winning lottery ticket. That means that I have no chance of winning tomorrow. No, you have just the same odds of winning tomorrow. Or I didn't win the lottery today, so my odds are going to go up for winning the lottery tomorrow. I'd hate to tell you, your odds are always the same. You have the same probability of getting that winning lottery ticket. So here we go. My first child had this. That makes it a 1 in 4 chance. What was the odds that the second child would have this? Same genetic composition. Well, again, it's a 1 in 4 chance. But now, the odds of having them both in a row is 1/4 times 1/4, which is a 1 in 16 chance, which, by the way, both of my children do have blue eyes. All right, so we're saying 2 recessive traits, though. These are unrelated traits. So, for example, Mendel crossed tall pea plants with tall pea plants, and then with the F1, he crossed them again. And what are the odds of ending up with the recessive trait of 2 heterozygous pea plants and getting that short pea plant? Well, we have capital T, that's tall, capital T, lowercase t, tall, capital T, lowercase t, again tall. And now we're going to have our 2 lowercase t's, and this is our short. It's a 1 in 4 chance of being short, right? So now, green pod is dominant over yellow. So again, we're going to take the F1s, capital G, lowercase g, capital G, lowercase g. And here we go, capital, and then capital lowercase, and capital lowercase, and then lowercase. So what we want to know is, what are our odds of being this lowercase t, lowercase g, lowercase g? What's the odds of ending up with that? Well, the odds of being lowercase t was 1 in 4. And the odds of being lowercase g, again, was 1 in 4, right? So that comes out to 1 in 16. Now, we could actually do this for all the traits. Like, for example, capital T something, we don't care, just being dominant, and capital G something, what would be the odds? Well, remember, it was 3/4 for this. Oops, that's incorrect there. Let me fix this. All right. This is a question mark. All right, so capital something, What were the odds? Well, remember, we had a 3/4 chance for being capital T, or capital T, lowercase t. And we have a 3/4 chance, capital G something, right? So that's 3/4 times 3/4. Basically, to be tall and green, that is a 9/16. I'm doing the math kind of backwards here. I'm going from right to left. Sorry about that. That's 9/16. Okay. Now, what if we wanted to be capital T something, so that's tall, but yellow, which has got to be lowercase g, lowercase g. Well, this was 3/4, right? And this was 1/4. What's 3/4 times 1/4? 3/16. All right, so let's flip this now. What about lowercase t, and a capital G, lowercase, and then something, sorry. It doesn't have to be lowercase, capital G something. Well, 1, 2, 3 of them were capital G, so that's 3/4, right? And lowercase t, and the last one was only 1/4. So again, we take 1/4 times 3/4, and we get 3/16. And if we add all of these together, 9 and 3 and 3, all right, that gives us 15, and then our 1/16 right here, that gives us 16. So that's 16 total possible outcomes, and that actually makes sense there, okay? So basically, if we cross two traits that are heterozygous, we should end up with a 9:3 to 3:1 ratio. That's a lot of math and a lot of work just to figure this out, right? We can actually do this on what's called a dihybrid cross, where we look at two traits at the same time. And so this is actually what the Punnett square would look like. And it's a 9:3 to 3:1 ratio. Basically, Mendel ran this cross. And he said, if these traits travel together, we're going to end up with a 3:1 ratio of yellow round to green wrinkled. Because these are both dominant traits and these are both recessive traits, and they travel together. Or if they travel independently, if they independently assort, which we've talked about before, independent assortment basically means just because you have blonde hair doesn't mean you're going to have blue eyes, and vice versa. They're traits that have nothing to do with each other. They're found on different chromosomes. And that's the same thing here. These traits are found on different chromosomes. And so what Mendel actually did find is that it did come out to a 9:3 to 3:1 ratio. And so this is what happens. So basically, we take a tall pea plant crossed with a short pea plant, and it has green pods, and we cross it with yellow pods. What are we going to end up with if it's homozygous for both traits? I'm actually going to skip over this, but you could work this all out on this Punnett square. I'm going to skip to the F1 generation just because it's mostly just to save you some time. Okay, so how do we figure out what goes in each box? Remember, we want one trait from the T's in each box and one trait from the G's in each box. All right, so this is the male parent and this is the female parent. We need to cross this. Now, if you are in a certain level of math, I believe it's at least Algebra 2, you should have learned about the FOIL method, where you take the first, outer, inner, and last letter. I'm sorry, it's number, I believe it's for binomial equations or something like that. But we're going to do the same thing here. We're going to do the first, outer, inner, and last letters of these combinations, because these letters they don't impact each other. And that's another important thing, is to remember, keep the T's together, keep the G's together, keep them separated, okay? We're just looking at two traits at the same time, and instead of doing two different Punnett squares and then multiplying everything out, we're going to do both of them at the same time. So let's do this one up here first, okay? The top one. All right. So we're going to take the first T and the first G, which is going to be capital T capital G. All right, I'm going to change pen colors here, I think. I hope. All right, pen color. Let's go with green. All right. Now, the outside T and the outside G, we're going to find the ones on the outside of each. So this is going to be capital T and a lowercase g, because that lowercase g is found on the outside. All right. Let's go with, now we're going to go inside. So the inside, nope, don't want that color. The color is way too similar to the last one we just did. All right, let me see here. Color. Sure, let's try this. All right, so we're going to go with the inside T and the inside G, which is going to be the inside T was a lowercase t, the inside G was a capital G. All right, and then I guess we can try yellow here. Let's see what yellow looks like. We're going to go with the last T and the last G, which would be lowercase t and the lowercase g. Okay? So, first T, first G. Those are the red marks. Then the outside T and outside G, those were the green marks. The white marks were the, I'm sorry, that was the outside T and G was the green marks, T and G. The inside T and the inside G were the white marks. And then the yellow marks were the last T and the last G. Okay. Now, the mother is exactly the same. All right, so I'm not going to color code this one just to kind of make it simple. So capital T, capital G for the first T, first G. If it helps you to make these marks, go ahead. Then the capital T, lowercase g for the outside T and outside G. The inside T and the inside G. Inside T, inside G. And then the last T, lowercase t, and lowercase g. Last T, last G. All right, so I got all the possible combinations. Okay, now this is where it's important to pay attention. All right. This is where students make mistakes. They start mixing their T's and G's. No, treat these like two separate traits. So, the first thing we're going to have is a capital T from the top and a capital T from the side. Then we're going to have, so that, put the T's together. That's our first trait. The G's have nothing to do with these T's, so just leave them alone for right now. So what is this? Tall? Short? This is tall. And now, mix the G's. Capital G, capital G. And so that's and. That's tall and green. Both the dominant traits, right? Since they're both dominant, we're cross heterozygous's, you should be predicting that we should see a lot of these, and we are going to. But we're just going to put a tally mark there to keep track of how many tall greens we've had. So that's tall and green. All right, so capital T from the side, capital T from the top. That's tall. Capital G, lowercase g. Again, the capital G from the side and the lowercase g from the top. So that's tall. Capital G, lowercase g tells us it's green. Got another one. All right, capital T, lowercase t. That is tall or short. It's tall. Capital G, capital G. All right. right, that's green. So again, tall and green. Remember, figure out the tall, then figure out the green. Capital T, lowercase t, tall or short? It's tall. Capital G, lowercase g, green or yellow? It's green. So first row, and I miscounted somewhere. That's only our fourth mark, so I went one too far. All right, so we got 4 so far for the talls. So that's why it's important to put tally marks and pay attention to what you're doing. Obviously, I had made a mistake there. All right, so capital T from there. Maybe just check when you're done at the end of each row to make sure that you've added 4 more. Capital G, lowercase g. So capital T, what is that? Tall or green? It's tall. Capital G, lowercase g. Tall or green? It's green. There's our 5th one. All right. Capital T. What is that? It's tall again. Lowercase g. Ooh, hey, something different. That's tall, right? But lowercase g is something different. It's yellow. I am not writing out the whole word yellow, sorry. I'm just abbreviating it. All right, tall and yellow. All right, so capital T, lowercase t. Capital T over here, lowercase t over there. Capital G, lowercase g. And I'm always putting the capitals for each pair first. All right, doesn't really matter other than just to get you thinking correctly. So this is, what's capital T, lowercase t? It's tall. What's capital G, lowercase g? Green. So that's tall and green. Capital T over here, lowercase t from up there. Capital T, lowercase t, tall or short? It's tall again. Lowercase g. Green or yellow? Hey, it's another yellow one. So that's tall and yellow. So at this point, we have a 6-to-2 ratio that can be simplified as 3-to-1. Yay! Exciting. We're done with 2 rows. Only 2 more to go. We're halfway done with this problem. So a capital T and a lowercase t. Tall or short? It's tall again. Capital G. Green or yellow? Green. All right, now we're up to a 7-to-2 ratio. Capital T, lowercase t. Tall or short? That's tall. Capital G, lowercase g. Green or yellow? Oh, too many G's there. All right, there we go. All right, capital G, lowercase g is green. That's tall and green. All right, 5, 6, 7, 8. We got an 8-to-2 ratio. That's a 4-to-1. All right, lowercase t. Tall or short? Hey, it's our first short plant. Yay, short. All right, so they're short. And they're going to be green or yellow? Capital G. That is a very funny looking G. Capital G. So that is short and green. All right. Lowercase t. If you can't tell by my quality of writing, my hand is getting very tired. So lowercase t, is that tall or short? That's short. Capital G, lowercase g. Is that green or is that yellow? That's short and green. There you go. Capital T, lowercase t. All right. So now we're on the last row here. Right now we've got an 8-to-2 ratio. That's a 4-to-1 ratio. So capital T, lowercase t, that is tall or short? It's tall. capital G, lowercase g, green or yellow. That's green. All right, 9, capital T, lowercase t, tall or short. It's tall. Lowercase g, green or yellow. That's tall and yellow. So that's 3. We got 9, we got 3, all right. Lowercase t, tall or short. That's our short. Capital G, lowercase g, green or yellow. Short, and that one's green. All right, lowercase t and a lowercase t, lowercase g and a lowercase g. That is now short and yellow. So we end up with a 9 to 3 to 3 to 1 ratio of tall green to tall yellow to short yellow. Sorry, to short green to short yellow. So sorry, let me do that again because I goofed it up. 9 to 3 to 3 to 1. Tall green to tall yellow to short green to short yellow. So back to our original question, what are our odds of having a probability of receiving 2 sets of recessive alleles? 1/16, 1/4 times 1/4. So what would be the result of a cross between 2 pea plants heterozygous for both traits? There you go. That's how you do these problems. Now, not to overly freak you out, but when you are looking at stuff for eye color, I'm going to actually skip over this epistasis stuff, which we'll talk about tomorrow. Again, you can see the Punnett squares are kind of similar, but it's the way you interpret the result are just going to be a slight bit different. But here is our dihybrid trait, right? It's a 9 to 3 to 3 to 1 ratio. Understand, with eye color, we're looking at 2, 3, 4 genes, maybe even more. Now, just with 4 genes, look how big and cumbersome the Punnett square gets. Notice they actually line them up like this, so this is capital, 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 capital. But like down here, we'd have capital, lowercase, lowercase capital, lowercase capital, lowercase capital. So this thing would actually be all 4 dominant traits, but they lined them up and down just to kind of make it simpler. You can do these types of problems. I will never make you, and actually I don't even know that you would see this until you got into like a college level genetics course, maybe even graduate level, a college level genetics course. I think there was a bonus problem I had in my genetics course for this. They tend to focus more on the DNA structure and the genes and such. I mean, yes, you do some Punnett square stuff, but you have computers for doing these types of things, right? So anyway, so when you talk about eye color, this is actually much more. You have genes A, B, C, and D all impacting eye color, and now you can see why when somebody asks me a question about, like, you know, what are the possibilities for different eye colors, or what are the genetic combinations that my parents could have had, and I kind of skirt the issue and say I'm oversimplifying it, because trust me, no one wants to sit through a lecture where you fill out one of these types of Punnett squares. Okay, so for today, what are you going to do in terms of homework? All right, and so let me go ahead and open up your worksheet. I'm not going to have you do all of them, okay? So this is the first page. Number 1 and number 2, you do not need a Punnett square. All you are doing is telling me, basically you are foiling the problem to give me what the sperm could contain or what the egg could contain. So you use the foil to determine the possible allele pairings. And then actually it says, and then cross a male and female guinea pig. Just foil for this one and for this one. And then I want you guys to go ahead and do number 3. This is going to require a Punnett square. If you want to do your Punnett square by making a grid in a Google Doc, that would be fine. And then numbers 1 and 2 down here, do these two, the ones with the eye color, okay? So, anyways, that cuts down a good chunk of it. Again, just FOIL this to tell me what would be in the sperm and egg cells. So basically, how would you fill out the Punnett square? You don't have to fill out the full Punnett square. Don't do part C for each of these. Do number 3. Do number 1 and 2. Do these two right there. All right? Okay. Anyways, have a great day.